This lecture looks in a little more detail at the light reactions. If you want to see the light reactions in the context of all of photosynthesis, there is a photosynthesis overview lecture that you can watch. So what is the purpose of the light reactions? Basically, we're going to use light energy, big E is energy, to make ATP and NADPH, two molecules. I'm going to mark these with the colors that I'll use for them during the rest of the lecture, and I try to keep consistent between other lectures as well. Use light energy to make ATP and NADPH. Why? To run the next part of photosynthesis called the Calvin cycle. Okay, so I'm going to draw the thylakoid membrane. If you don't know what I'm talking about, go watch the overview of photosynthesis lecture. And I'm going to draw some structures embedded in the membrane. These are complexes of proteins and other molecules. Draw this in a different color here because it's embedded in the membrane. It's just part of the ATP making process, which I usually draw in orange. So this is the thylakoid membrane, and then we have these protein complexes with a bunch of pigment molecules embedded in them. So that's what these guys are. Pigment molecules. Why does that matter? It matters because they can absorb sun energy. So let's draw some of that, or light energy. That's what goes into the system. That's the input of energy. So we have the light energy going into the system. It bounces around from pigment to pigment until it gets boosted up enough, the light energy, to boost the electrons to a high energy state. So I'm going to call these boosted electrons. The lowercase e with a minus for electrons which have a negative charge. So we got these boosted electrons hanging out there. Those electrons are going to get passed over to this chain here of complexes, and it gets passed from the chain from one to the next to the next. And this is a chain of electron passing complexes. It should, if you've done cellular respiration, look very familiar to you. It again is an electron transport chain. That makes sense. So this is the ETC, or electron transport chain. A different chain than they have in cellular respiration, but it does exactly the same thing as far as function goes. So it's these complexes. And what happens when these electrons get passed? Well, they release a little bit of energy. And so what we can do is to use that energy to do transport. So we're going to be pumping hydrogen ions, or protons, just like in cellular respiration, across the membrane into an area where it's already crowded. So there's lots of hydrogen ions or protons inside the thylakoid, and I'll draw a thylakoid little picture in a minute. So there's lots in here already, which means the hydrogen ions aren't going to want to go into. They want to stay all spread out. But then you're using the energy of the falling electrons to pump them into this crowded space already. So if we're just thinking about the thylakoids here, it's that stack of membranes. And so we're taking the hydrogen ions and pumping them into the inside of all the stacks, like in the middle of the pita bread. Filling that up and just jamming more and more and more hydrogen ions into the middle of that pita bread there. So this is going to end up being high hydrogen ion, H+, or you can call it a proton. Concentration on the inside, which means it has lots of potential energy. Remember that energy that's stored, it has potential to do work. Lots of potential energy in the system. And we'll see what happens with that in a minute. And let's mark this off because this is not part of the thylakoid. This is just showing it from a kind of a larger, like a zoomed out perspective. We're zoomed in on this top picture. So what happens with our electrons after they've fallen down this electron transport chain? They get passed to this other complex of proteins and pigments, and that complex is also getting light. And so it's bouncing the electrons around until they get boosted. Just like the other photosystem. And after they get boosted, they can be taken away from the system. So this is where we have our NADP plus picks up those electrons and leaves as NADPH. So the NADP plus is the version without electrons. Let's do it in a different color. Let's write it with purple. Okay, and you can think of this as the empty cab. And the reason I say cab instead of rideshare is because if you've never ridden a cab before, which is highly probable, when it has its light on, then that means it's empty. So I think of this little plus as like the light on, meaning empty cab. I'm here, I can pick up more passengers. It picks up its electron passengers, and then it's a full cab, so its light's gone off now. It picked up the hydrogen too, but there's no more light on this. So this is a full cab with the electrons in it. And that matters because it's going to take those electrons to the Calvin cycle. Because as we mentioned, that's the point of these light reactions, is to make stuff to make the Calvin cycle. So CC is Calvin cycle. So now we've made our NADPH. If we think about this system, it needs to keep getting electrons in to keep flowing, right? Because you're taking electrons to the Calvin cycle. Well, how are you going to get new electrons to put into this photosystem here, to put into the complex with the pigments? Where are we going to get those new electrons from? Well, that's when water comes into play. So water, H2O, actually brings in new electrons and drops them off into the pigment complex. So it keeps feeding electrons into the system. So water is your electron source. And once it's dropped off the electrons, it leaves as oxygen. So the hydrogens fall off, and then you've just got the oxygen left. So then you get two of those put together to make an oxygen molecule. And that's gas. And this is a byproduct, not the point of photosynthesis, but just, it happens to be formed by it. And that goes and diffuses out of the cell, well, first out of the chloroplast, then out of the cell, and then it could be used for cellular respiration by either the plant itself or by other organisms. So we made our NADPH to ship our electrons to the Calvin cycle. We're getting more electrons from water, so we make oxygen as a byproduct. The last thing we need to do here, as you can see, is to make some ATP for the Calvin cycle. And that's when this orange complex comes into play. This is your ATP synthase. Same actual exact molecule that was in cellular respiration. It's an enzyme that makes ATP, an ATP synthesizer, ATP building enzyme. So how does it work? You get these hydrogen ions and they flow through the ATP synthase, and that spins around and can basically take the ADP, add another P to it. The D stands for di, which is for 2. So it has 2 phosphates to begin with, 2 Ps. You add another P to it, and then it has 3 Ps, so that's ATP. Remember, this is a very high energy molecule, ATP. So that's good, and we can send that to the Calvin cycle so it can be used for building the carbon compound, the sugar, the organic molecule, however you want to say it. So there's our 2 products, ATP and NADPH. So if we look at it in our zoomed out picture of our thylakoid stack, we have our ATP synthases in the membrane. Lots and lots and lots of them. You know, I always just draw one in my pictures, but there's tons. And those hydrogen ions are flowing out. And as that's happening, they're able to make the ATP from the ADP and the phosphate group. And to remind us, how did we get those? How did we get the protons to get shoved inside there to begin with? We use the electron transport chain to pump, that's an active process, pump the hydrogen ion / protons into the thylakoid space. So you do active transport to pump it in there, to get it crowded, to let it flow through, to use that energy of flowing to make ATP. And that's the very basic idea of the light reaction part of photosynthesis.